Good morning, everybody. Bring up the chat. All right. So if memory serves me correctly, and I actually cheated because I checked the YouTube video just for class. We are at this point, all right? So we've already covered LAC operon in detail. And we've started on the trip operon, but we haven't really gone on to the fine control, right? So just to recap, the broad control for the trip operon is all about a negative feedback from tryptophan, right? So when tryptophan is present at high enough levels, and that's kind of key, it binds to the aporepressor TRPR, and that binds to the LAC TRPO operator, right? Shutting off transcription completely. Now that's very much the same as the LAC-I protein being on the LAC-O operator, right? It's a very much an on or off deal, right? And so there's something kind of an in-between ground where bacteria can kind of fine tune that level of trip operon expression. Because as you can imagine, in normal conditions, tryptophan's constantly being consumed by translation protein synthesis as it's being made by the products of the trip operon, right? So you have that kind of constant homeostasis, right? That balancing act between the two. And that's all about attenuation, right? So attenuation is, I don't know, best put it as cutting short, right? If a signal is attenuated, it's cut short before it's finished. If your sentence was attenuated, which would be kind of a weird use of the word, but you wouldn't have finished the end of the sentence. And so the key to this, where are we, is this region here. It's the leader region. And that's how trip operon expression is attenuated, like the fine control, essentially. And so the point behind this, and this is actually a very tricky thing to get, right? So I'm kind of learning every semester, obviously, how to explain this well, but also from the other section too, right? So something came up there which should help today. And so basically the trip leader region is a short stretch of mRNA, which is really part of the polycistronic mRNA. Right, so remember, all of this leader region and all of the structural genes are transcribed as one large mRNA. Really important to get that, right? That little stretch is also translated into a essentially unused polypeptide. Right, so the structural genes code for the enzymes that do stuff, right? The leader region codes for a very short polypeptide that's sole purpose is to be part of the mechanism that senses tryptophan levels. Okay, so question to the class. What's, other than polycistronic mRNAs and operons, what's another key characteristic of gene expression in prokaryotes compared with eukaryotes? What's one of the key characteristics? Think about where transcription and translation occurs in bacteria. Just throw some ideas out there. Well, I have cytoplasm, I guess. I mean, it's just one large compartment in the bacterium, right? They don't have organelles. So if they don't have compartments or organelles, where does, sorry, I'm having a snack because I didn't get a chance to eat before lecture. Where does translation and transcription take place relative to each other? Different places, same place. Which one? Same place, great. What about time? Is the mRNA produced first? I'm sorry, don't know what that was. At the same time, right? So transcription and translation happen at the same, in the same place at the same time. So as soon as a piece of mRNA is long enough, translation begins. That is everything to how the trip operon is attenuated. All right, so let's come at it from a different direction. What's tryptophan used in? What is tryptophan? What is tryptophan a group, part of a group of? And they all have different R groups and different properties, but they're all used in what process, what part of gene expression? Give you a clue. Name ends with an acid. No, it's not nucleic acid. What is tryptophan? It's an amino acid. Great. Thank you, Jesus. Right. So amino acid, tryptophan is amino acid. Amino acids are used in translation. And the leader region encodes a short polypeptide that will be translated as the leader RNA is being transcribed. Right. So the coupling of transcription of the leader sequence or the leader mRNA and translation of the leader mRNA are, pardon me, how tryptophan levels are sensed. So if tryptophan levels are going to be sensed by translation of the leader region, what codons would you expect to find in the leader region? Yeah, that's a good point. Start codon. Got to start somewhere, right? What else? If we're sensing tryptophan, tryptophan is amino acid. We want to sense it. Well, the anticodons on the tRNA, right? What thing are we trying to sense? Which amino acid do we care about for the trip codon, tripoperon? Oops, just told you. Just look a little bit further up in the chat. What amino acid is the tripoperon involved in making? Come on, peeps. What amino acid is the tripoperon involved in making? Where does it get its name from? Tryptophan. Thank you, Sayed. Exactly. Right. So what codons would you expect to find in the mRNA that's involved in sensing tryptophan? Tryptophan codons, right? Exactly, trip codons. So the bacteria is going to be sensing tryptophan levels directly based on how much of it is available for protein synthesis. And the protein we're going to synthesize is the one in the leader region. Okay, so now we've got some more details. Let me just cover this up because it gets a little bit hairy. Okay, so over here on the right, about where Sayed's head is on my screen, is the start of the structural genes, right? Trip E goes E, B, C, B, A for some reason. Right? And so we have this leader region, right? And it gets translated. It's really, really short, tiny polypeptide, right? That polypeptide doesn't do anything. Its sole purpose is to allow tryptophan levels to be detected. Okay. We've got 4 different regions, right? These 4 regions can pair with each other in different combinations. We have 2 different combinations, right? So one combination is 2 and 3 form a loop, right? So these 2 form a loop. And remember, transcription is a linear process. It sounds silly to say it, right? But region 1 is produced before region 2. Region 2 is produced before region 3. Region 3 is produced before region 4, right? So if these regions are able to do so, they will pair up and form a loop as soon as they are made. Okay, so if region 2 and 3 are produced and are able to pair before region 4 is produced, 2 and 3 will form a loop. It's not going to hang around and go, ooh, no, actually, don't really like you all that much, until 4 is produced and 3 and 4 can form a loop. No. As soon as 3 is produced, and if 2 is available, 2 and 3 will form a loop. Remember that linear process, right? 3 and 4 can also form a loop. Right? If 2 is not available. Okay, so we've got these two different possibilities. Okay, let's get this out of the way. No, that's the one I want. Here we go. So if loop 2, 3 forms, nothing happens. Transcription continues. And that's transcription into the structural genes, right? Oh, sorry, continues. And that's okay. So these different loops have different consequences. And we have two different choices. 2, 3 loop, if that forms, nothing happens. The structural genes will get transcribed and translated as normal. Now, if this is a slightly erroneous picture, but we're not going to worry about it right now. If region 2 is not available, right? And we'll talk about how it would not be available, right, in a little bit, like how these two different options are essentially chosen, right? So we have two different options. We're only going to get one of them, right? So what controls the switch between one versus the other? We'll get to that in a little bit. I just want to kind of lay the groundwork of what those two different options are. So if region 2 isn't available, and it's not actually because it's bound to region 1, I don't know where they get this picture from, but it's wrong, right? Well, it's not entirely correct, let's put it that way. Region 3 has nothing to pair with until region 4 is transcribed. All right, so remember, I was telling you, if there's something that region can form a loop with, it will do. Right, so the only reason why you don't get region 2, 3 is if region 2 is not around. So region 3 kind of just hangs out in the breeze waiting, right? And then when region 4 is transcribed and region 3 is still available, the 3-4 loop will form inside the RNA polymerase. Sorry, I can't always see what I'm typing. There we go. And that will kick it off the DNA and ending transcription. And that essentially attenuates, right? That's where the name comes from. It cuts short transcription. So even though you can make one large polycistronic mRNA, including the leader region and the 5 structural genes, if loop 3-4 forms, that's stopped kind of over here before that whole mRNA can be transcribed. And also take note of the sequence of U's. That's not US, U's. I'll probably better put an apostrophe next to it. Those are there to pause RNA pol. Right, so if you remember about intrinsic termination of transcription from way back when, section 3, that we're talking about it. This terminator loop and the poly U are the mechanism of intrinsic termination, right? The U's slow down RNA polymerase while region like this terminator loop forms within the channel of the RNA polymerase. And if you compare the alternative where 2-3 form, there's still that, you know, series of U's, but the loop forms outside of the RNA polymerase. It forms away from the RNA polymerase, and so it doesn't disrupt transcription. Okay, I'll have some coffee and let you think about that. Have a cogitation on it and see if there's stuff that doesn't fit, make sense, jive, that sort of stuff. All right, anybody got questions or needing clarification?
I have a question.
Go for it.
So 2 and 3 and 3 and 4 can form a loop. So why can't 1 and 2 form a loop? Like, isn't that what on the picture on the left? Isn't that the little like bottom part? Would that be considered a loop? Or
well, this is if the RNA was kind of free to do its own stuff, which it isn't, because there's a ribosome bound to it. And so the reason why I'm a little irritated by this particular image is because it's not actually 100% accurate for what's going on. This one is, right? The one on the left is not. And the reason being is that there's actually a ribosome bound to region 2, right? Because remember, transcription and translation are coupled. And so as soon as this ribosome binding site becomes available, a ribosome will bind to it and begin translation. And so region 1 and 2 are really there to dictate essentially which of these 2 options you get. Really just region 1 actually in many ways, but we'll get to that in a second. Did that help?
yes, it did. Thank you.
Cool. How do you mean, like this, like these various weird secondary shapes, Michael?
Yeah, just right there.
Right, so these are, I don't know if it's either artistic license or it's just part of the various secondary structures that these things can form, right? And so you don't get, like, for example, region 2 and 3 have a perfect pairing, but region 3 and 4 and 1 and 2 don't quite have a perfect pairing. So you have this little bit of a kick out on both of those. But in the grand scheme of things, in terms of what we're interested in, that doesn't really matter, right? All that you really need to kind of focus on is which of these two options forms, because these are the decider as to whether or not transcription occurs or does not occur. That help?
Yeah, thank you.
Yeah, I mean, RNA can form all kinds of weird and wacky shapes. So would not be surprised at all if these actually reflect exactly what it looks like. Okay, so these are the two different options, right? And they dictate whether or not transcription proceeds into the structural genes or terminates on the 3,4 terminator loop. So now the question is, how do you choose? How does the bacteria kind of, well, not choose because it's not a conscious effort, right? How is that switch made between transcription continuing and transcription terminating, right? And that comes down to solely these two trip operons here. These are the sensor, essentially, of tryptophan levels. So what's going to happen if you have no tryptophan in terms of charged tRNAs? Are you going to have any tRNA with tryptophan attached to it if there's no tryptophan available? No, right? Sorry, kind of jumped ahead there. Should let you answer. So what these trip operons are sensing is the availability of tryptophan for translation. So now we're going to go back to the coupling of transcription and translation. So this is a really nice, I actually like this image a whole lot better. Just going to zoom it in, and I'm going to blank off bits so we can walk through this kind of bit by bit, right? Because it's a lot to take in, and I want to make sure that you kind of follow it all. Okay, so transcription begins, right? And this is conditional on there not being enough tryptophan to induce the tryptophan repressor to bind. Okay, so we've already got past the whole on-off deal of transcription and control, right? So transcription is on, right? There's not enough tryptophan around to turn it off. There may still be some around, but there's not enough to turn it off. Okay, so transcription begins, and region 1 is transcribed first, obviously, right? Because it's done in an order of number, thankfully. As soon as that RNA polymerase has moved far enough along, a ribosome will be able to assemble on the start codon, right? And it will begin translating region 1 while region 2 is being transcribed, right? So these two things are going in lockstep, right? They're not coupled, but they're occurring at approximately the same rate. Okay, so this is a very linear deal. Region 1 transcribed first, then ribosome 1 binds to it as region 2 is being transcribed, right? So depending on what happens with the translation depends on what kind of, not what gets transcribed, but what loop forms. Okay, so what do you think will happen if there is no tryptophan available or very low tryptophan when this ribosome gets to these two? Da. I'm sorry, my phone was talking to me for some reason. What do you think will happen when this ribosome reaches the trip codons if there's no tryptophan around? What's going to happen to the process of translation?
Is it stalled?
Exactly, it cannot continue because there's no incoming trip tRNA to allow translation to continue. Yeah, so it doesn't just slow down, Jonathan, it actually stops, right? It stops until there is a trip tRNA available to come in, and there's actually needs to be 2 of them, right? So that you have to have at least 2 trip tRNAs around for translation to continue. Okay, so ribosome is going to be stored on region 1, like so. So if the ribosome stalls on region 1, okay, the RNA polymerase continues transcribing the leader region, right? It doesn't stop, at least not yet. So if region 1 is not available for pairing, region 2 will be. Okay. So what's going to happen when region 3 is transcribed? What are you going to have form?
I have a question.
Shoot, go for it.
You said, so because there's no, not enough trip operons that the ribosome couldn't
Trip-
continue, right?
Trip tRNAs.
Oh, sorry, trip tRNAs. It couldn't continue.
Correct.
So if, how is 2 going to be ready if the ribosome hasn't gotten there yet?
Okay. So that's a really good question, because essentially what the ribosome is doing is blocking different regions from pairing. So regions of the mRNA, because it's physically on the mRNA. Right? So if the ribosome is physically on region 1 of the mRNA, right, region 2 could be just flapping around in the breeze, but it's not going to be able to pair, right? Because region 1 is covered.
Okay.
Right. And so if the ribosome stays there, region 2 is still going to be flapping in the breeze, as region 3, I don't have enough hands, sorry, as region 3 is transcribed. Does that make sense? So region 1 is still being covered because the ribosome is paused there, it's stalled. But the RNA polymerase is continuing along merrily and oblivious, transcribing the leader region. Does that make sense?
Yes, thank you.
Okay, so as soon as region 3 is transcribed, now 2 and 3 are able to pair, as Cynthia said. And you get, oh, let's see if I can scroll down a wee bit. You get this, right? And because region 2 and 3 have formed, you can't pair region 3 with region 4. Right now, region 3 is unavailable. Okay, so the ribosome stalling on the first region, essentially, like as the outcome, stops region 3 and 4 from pairing. So if you kind of think through in terms of this follows this, follows this, follows this, right? Which is kind of really essentially what it's all about. It's just a kind of like a logic kind of equation almost, like this has to follow after this, right? If the ribosome stalls because there's not enough trip tRNA, region 2 is available. So when region 3 becomes available, it forms a loop. and remember, region 3 is produced before region 4, which is what you're seeing here. So region 4 is still being transcribed. It's not available when this pair forms. So if region 3 is already paired with 2, as region 4 becomes made, it has nothing to pair with, right? Because region 3 is already paired up. Kind of like, you know, teens at a prom dance kind of thing, right? That's actually a really terrible example. Sorry. I've got teenage daughters, which is why I think about this all the time. Anyway, let's get past my angst of being a father with teenage daughters. And so essentially that loop is far enough away that the RNA polymerase is not affected by it. And so the RNA polymerase just continues on transcribing the structural genes. So let me just type that sequence of things down, right? So not enough trip tRNA. Equals ribosome stalls on region 1, because that's where those trip codons are. Ribosome stalling on region 1 equals region 2 available. Region 2 available equals pairing with region 3, sorry, when it gets transcribed. And then the region 2-3 loop equals region 3 unavailable for pairing with region 4. Equals no terminator loop form equals transcription continues. Right, that's essentially the series of events in a very linear way, right? Because this is a linear process. Region 1 will be transcribed before region 2. Region 3 will be transcribed before region 4, and so on. So low trip tRNA equals, transcription of the trip operon equals more tryptophan. Sorry, typing's not working today. Right, so if there's not enough tryptophan around for the ribosome to continue translating the leader sequence, you get transcription of the trip operon and you start making more tryptophan. So what do you think is going to happen when tryptophan levels start going up? What's going to now become available?
Is it region 1?
Well, the trip tRNA is going to become available. So what's going to happen to that ribosome on region 1? Is it going to stall? Or is it going to continue?
It's going to continue.
It's going to continue, right, exactly. Now let's move on to the next one. Zhangnats. Let me just.
You might have already covered this, but that last square says the attenuator terminates transcription.
Mhm.
What's the attenuator?
So that's essentially the terminator loop. That's the 3,4 loop or structure. So it forms inside the RNA polymerase and it physically forces it off of the DNA. So essentially this is intrinsic termination of transcription, right, which we learned about 3 or 4 PowerPoints ago. So attenuator, terminator, kind of interchangeable names essentially. Loving all the questions though, this really helps. Okay, so now now we've got expression of the trip operon, right? Produces the enzymes that turn chorosmate into tryptophan. Tryptophan levels start going up. Okay, so we're going to start the same way again, right? So there's not enough tryptophan to shut off the trip operon completely, right? We're looking at an intermediate level essentially. So transcription is occurring, right? So transcription begins, here's RNA polymerase, transcribes region 1, right? So as soon as region 1 is available, a ribosome binds to it, right? And begins translation. And region 2 is being transcribed. So again, we've got the same thing happening. Okay. However, now there is sufficient tRNA. Okay, so this ribosome does not stall on region 1. It does not stall, but continues into region 2. Okay, so region 3 is not produced yet, right? It's not been transcribed. So if the ribosome is moving into region 2, it's no longer going to be available for binding to region 3. Right, kind of comes back to that physical blocking, right? So the ribosome has done region 1, now it's moving on to region 2, and so it's physically going to prevent region 2 from binding to region 3. And so if the 2-3 loop doesn't form, what loop are you going to get? The 3-4 loop. And the 3-4 loop comes with those U's, which will slow down RNA polymerase, right? It doesn't stop it, it just slows it down. And if that 3-4 loop forms, then the RNA polymerase is kicked off and no transcription of the structural genes occurs. How's that going? So now you can see how this is such a cool example of that principle of gene expression in prokaryotes, right? The fact that they are coupled. And this only works in bacteria, right? It's not going to work in eukaryotes because in eukaryotes, they're not coupled. Okay, does anybody have any questions? No? All right. Okay, so make sure you go back through this because it's, as you can see, not trivial, right, to get. And there's a lot of stuff going on, but as long as you kind of do the whole tortoise versus hare thing and just work through, okay, what do we have in this context? What happens now? What does that result in? And what's the outcome? It should be pretty straightforward. Okay, so let's change gears. Now we're going to look at eukaryotes. Right, so again, if you have any questions about any of this stuff, bring it to class on Tuesday. So remember, we have class on Tuesday, right? We don't get the week off, unfortunately. So we'll do that, but we will not have lab next week. Right, we have lab this week. We do not have lab next week, okay? So if you're in the second kind of grouping of lab sections, you'll be coming in the week after Thanksgiving, right? That will be the last lab, or last week of labs, essentially. Okay, so in many ways, like prokaryotes are, they're both elegant and simple, right? So and porks? No, prokes. We don't want porks. Those are eukaryotes. And they have spines too, right? So in prokes, most control of gene expression occurs at the level of transcription. In eukaryotes or euks, it can and does occur at any level between, I guess, chromatin state and protein processing export. And so as I said to the other section, you know, this is, it's kind of by far my favorite, you know, subjects that I teach in genetics. You know, the transgenic stuff is pretty fun as well. But it's also one of the hardest too, because there is a lot of stuff going on. And it can quite easily be overwhelming, like lots of details, lots of this and that. So try and keep in mind the bigger picture, right, of what is going on. And really the big picture, let's see if I can, Yeah, it's still open. Awesome. So this is a picture from my DevBio class. And I will add this to the PowerPoint. I meant to yesterday, just ran out of time, and upload it because there's a cool story about this as well. So this is an alligator embryo, right? I believe it was done using taken of fluorescent protein reporters, maybe immunohistochemistry. I'm not actually 100% sure. I'd have to read the article again. But essentially, this is an alligator embryo fairly early on, right, in development. I love talking about development because it kind of epitomizes essentially what we're going to be talking about in terms of control of gene expression, right? So every single cell in this embryo has the same instruction set. Every cell in your body, apart from red blood cells, because they don't have DNA, or at least they don't have genomic DNA, every cell apart from red blood cells in your body has the same instruction set. And yet they are very, very different, right? And so how does that occur? And that's essentially what we're going to be talking about in this section. And so I love showing this particular image because I'm a bit of a microscopy nerd in many ways. You know, that's been a big part of my, you know, scientific life, essentially, is taking pretty pictures, cool pictures. And this is one of my favorites because this is a multicolor laser scanning confocal microscopy picture, right? So this is taken, so alligator embryos, you know, I mean, this isn't like a big saltwater croc that's gonna, you know, eat your dog or something, but this is still a pretty big sample, right? It's probably about that big by about that big, give or take, which in microscopy terms is huge, right? It's absolutely enormous, actually. So this is taken by shining a laser. Remember we talked about fluorophores in the Sanger sequencing lab? This is a collection of different fluorophores, right, which emit at different wavelengths when they're excited by different wavelengths. And so essentially you have a laser, it just scans back and forth like that. And then it does a different level and a different level. And you can actually slice a sample in, I think, as little as 1 micron slices, right? And so this image probably took, I'd say, several hundred slices easily. And that's done at each color, and then it's kind of smooshed together to give you a multicolor image. And so when you look at this, here's the brain, this kind of green thing. So green stuff, green labels the neurons. And so you can see the brain here, kind of think that, well, the brain should be up here, but this is actually where the future jaw is going to be. So you can even see these kind of bits here, right? These kind of, they look like 4 fingers, right? There's 1, 2, 3, 4. Those are the branchial arches, right? And so those actually kind of come together, you know, predator-like, from Alien vs Predator, come together to form your upper and lower jaw. And that's where the whole hair lip thing comes from, right? In humans, if those don't actually meet, you have a dividing line from about here, give or take, down where those meet together. So you can see the spinal cord, right, coming down, innervating, right? So these cells are enormously long, right? So they kind of start up here and can go all the way down to the tip of its tail. And you can see them innervating different blocks. Obviously that's the heart or developing heart, and these are called somites. And so they're blocks of cells which will become vertebra and back muscle. So a lot of the muscle along the back, essentially, and ribs and things like that. So these are distinct, and actually the identity of these different blocks is different. They are discrete. So the block at the top, right, this will become a, well, I guess in alligators, a cervical vertebra. Blocks down here will tell cells around them, you need to develop into arms, or in this case, legs, right? All of this is controlled by different patterns of gene expression and signaling, right? So these all have the same information. They have very, very different properties and different fates. These will develop into the back muscle and the spine. Cells around here will develop into the lungs and the gut, you know, brain up here, and so on and so forth, right? And so this is all about the control of gene expression. And that's why I love showing this picture, because it kind of absolutely gets to that kind of point that we're going to be talking about. And so this image, really, this is just looking at transcription. And what I'm going to spend the rest of the class period talking about are essentially the different levels at which this can occur, this control can occur. And this can occur at any and all of them, right? It just depends on what gene we're talking about and the context, right? You know, is this a gene involved in a process that needs to happen now in this part of the body or not? If not, it's not going to get turned on. If yes, it will, and how much depends on a whole bunch of other processes, right? So essentially, you can have signals which are intrinsic to the cell. So if the cell, you know, is struggling to fold its proteins properly, right? This is actually part of a lot of neurodegeneration diseases. It will trigger a particular pattern of gene expression that may lead to its own death. Cells can also be extrinsic, right, from outside of the cell, right? Telling that cell, you need to do this, you need to do that, right? This is what has to happen. And so essentially we have at the very, very end of this functional protein, right? That's the outcome, the output of gene expression. I mean, it might be a non-coding RNA too, but for most intents and purposes, we're going to be talking about proteins. And so how much and where and when of this protein you get, right, is a sum of all of these different steps, right? It's like an equation, essentially. You got this and this and this and this. This is your outcome. And it starts with the state of chromatin. And so, actually, can anybody remember the two different chromatin states and the open versus closed, which is which? Yeah, thank you, Jacqueline. Heterochromatin and euchromatin. Can anybody remember which one of those is the open or on state? It's funny, I often think of it in terms of the European Union when they let all those refugees in from Syria and the like a few years ago. So euchromatin is the open state. Obviously not so much anymore with current political and migrant crises in the European Union, but it's how I remember it anyway. Right, and so again, it's not that genes will be expressed, it's just that they're able to be expressed, right? heterochromatin is closed and off, right? So if the DNA which happens to span that bit of the chromosome that encodes that gene is heterochromatin, pardon me, you're not going to get gene expression. All right, it will be off. Right, so essentially switching between those two states dictates whether or not that DNA is available for a gene expression. Right, and quite often, you remember we talked about how gene expression is coordinated in bacteria by grouping all those different genes together to make an operon, right? So in eukaryotes, we don't do that, but instead, quite often you'll have many genes grouped into the same region of the chromosome. So if you need to make an arm, like that one, or that one, actually they get switched on at the same time, as you guess. There's a whole bunch of different genes which are going to be specific to limb development. Right, we've got innervation, not innovation, right? So nerves have to move in. We've got bones must be laid down. We've got muscle and so on and so forth, right? And some of those are specific to your upper limbs, right? These ones. And so those genes tend to be kind of grouped together. And so if you don't need to make any limbs, right? I've got two, don't need any more. Although, yeah, maybe sometimes it'll be handy to have an extra limb or two, right? But that whole program can be switched off. And so you don't have to worry about turning off or repressing individual genes. You just do the whole lot at once. Then obviously, which is really what this gets at, you can control whether or not those genes are turned on. So the control of transcription is carried out by the presence or absence of certain proteins. And those are called transcription factors, really the ones that turn on gene expression, and repressors are the ones that turn it off, which recognize, make sure I type that out right, key DNA sequences in the regulatory promoter. And so transcription factors here, these enhancers or activators, repressors, whether or not those are present and whether or not the promoter has the sequences that they recognize will dictate whether or not that gene is turned on. And then because, again, and this is another difference between prokaryotes and eukaryotes, and so kind of going back to this, right, just to kind of re-emphasize, genes in eukaryotes are off by default. So you have to have things to turn it on, those things being these activators. And transcription factors too, right? So that's one big difference. Also, in eukaryotes, we get a primary transcript, right? So this contains introns, which obviously prokaryotes don't have. And so controlling how those introns are removed, which ones are taken out, and if any exons kind of go with them, is also a way in controlling what is expressed. Right? And we'll talk about how you can actually have multiple different proteins produced from the same bit of DNA, from the same transcript, actually, which is pretty crazy. It's not a one-to-one deal like you have in prokaryotes. So we can control processing. I don't think transport to the cytoplasm is something that's really kind of a level of control, to be honest. If it's processed, then it leaves, essentially. But now we have that mature mRNA in the cytoplasm. Okay, now we can control two different things. One, whether or not that RNA or mRNA is translated, right? So we can control gene expression at the level of translation. We can also control how long that mRNA hangs around for. And so if we want to turn off gene expression or reduce gene expression, well, then all you need to do is degrade the mRNA. And now there's nothing to translate, right? So gene expression is off. So providing, we have stable mRNA, right? We'll talk about how degradation is actually controlled. And it's translated. Now we get our polypeptide. And so this is not the end product, right? So this is a kind of linear sequence of amino acids. It's not a three-dimensional functional protein yet, right? And so there's a lot of stuff. And so it's kind of funny because in many ways, when we talk about gene expression, right? Typically we stop here, right? But in eukaryotes, there's an awful lot more that happens after translation in terms of functional proteins, right? And actually in many ways, transcription and translation have absolutely no effect on the level of functional protein. It's stuff that happens after translation. That's called post-translational modifications. And there's a lot of different things that can go on, right? We can have cleavage, we can have add in things, we can remove things, right? We can dictate whether or not that protein hangs around, right? So proteins can be tagged for degradation, maybe because they're damaged, maybe just because they're not used anymore, right? In fact, in terms of cell signaling, right, which is something that I do a lot of work on in terms of research, as soon as a signal's been received, the thing that detects it is destroyed. Right, much the same as when you have a nerve impulse, right? You release neurotransmitter into the synapse, right? As soon as that nerve impulse is gone, you remove it, right? Because you don't want to keep firing those nerves, otherwise you'll be like jiggling and jangling around, right? So getting rid of protein, as with mRNA, is another way. And then just even getting that protein to the right place, right? So insulin is a great example. It's not actually a functional protein until it's been cleaved, and then until it's been released into your bloodstream, right? As it sits in your pancreas, it is a non-functional protein, can't do anything, because its targets are your muscle cells and your liver, right? And your pancreas is not next to your, well, it's kind of next to your liver, I guess, but you get what I mean, right? It's not available to do something. And so you can see there's so many different steps, right? And we'll be going through these kind of one by one by one. right? And so there's a lot of detail in all of this because these are complex processes, right? And there's a lot of detail even in the relatively, I'm not going to say simple or basic, but or even superficial, right? But I'm giving you the Cliff Notes version, essentially. Even in that, there's a lot of stuff going on, right? So keep in mind this bigger picture of, we've got this DNA, we need this protein. How do we get from one to the other, right? As we go through all of those, so it doesn't become just overwhelming. All right, that's enough for today. I'm gonna sign off, have some lunch, and I'll see some of you in lab, and we'll continue this on Thursday. All right, see you, everybody.